What's good, everybody? And welcome back. Welcome back to another video. And welcome back to my lab. Today we are doing something very different to what I've done before. And as you can tell, I'm not whispering because I have to use this little mic because I can't put the big one in here. And the little one isn't going to pick up my whispers with all the noise of the suction from the hood. But anyway, yeah, today we're in the lab. I need to make some feeder layers, which I'll explain to you what those are. But I thought that it would be cool to show you how I make them. And I thought I could also show you like my cells that are growing and stuff. I thought it'd be pretty cool. And yeah, so I hope that you enjoy the video. I hope that it's relaxing. Hopefully, I have no idea what it's going to turn out like because of the hood's quite noisy. But hopefully this will be relaxing and there'll be some nice sounds. Before I start as well, I mentioned in my last video and I did a post, but just in case you didn't see, we hit 5,000 subscribers, which is crazy, so thank you again. But I'm doing a Q&A for that, so if you have any questions, comment them on this video or on the post, or wherever you want. I'll find them and I'll answer them in that video. So I'll be doing that sometime in the next couple of days. So yeah, right. I need to do this first step kind of quick because there's like an hour-long, 3-hour-long incubation time. But basically, I'm going to do it. I'll talk through what I'm doing and then I'll explain what feeder layers and stuff are after. But yeah, I'm meeting a friend. I'm meeting a friend in a couple of hours, so I can't be too long. So, and it would be good to get the 3-hour incubation period started. So, I'll talk through it as I'm going. But basically, what I have here are some mouse embryonic fibroblast cells. And we basically use these cells, we grow these cells in these flasks, and then we do what I'm about to do now, which is, it's called mitotic inactivation. So we don't kill the cells, but we stop them from growing. And then we basically grow human epithelial cells on top of these cells. And the mouse cells, the feeder cells, help the human cells to grow. So what I'm doing now is basically turning them from the mouse embryonic cells, fibroblast cells, and I'm going to inactivate them to turn them into feeder cells. And so to do that, I need to add something called, a chemical called mitomycin C. And I basically dilute it in the normal growth medium and then add it on top of the cells and leave it for 3 hours. So this is the chemical. It comes as a powder and I've just dissolved it in water, I believe. I have to look up what I need to dissolve it in every time because different things are dissolved in different things. Anyway, mitomycin C. And this is 100 times too concentrated. So I'm going to add, this is like the growth medium. I'm going to add 8 mils of growth medium to each flask, which means I need to add 80 microliters of mitomycin C per 8 mils. So I've got 48 mils of medium in total, and I'm going to add, what's 80 times 6? Well, I'm going to add 480 microliters. So get my pet tips, and I'll set this to 160 microliters. And each one of these has 180 in, so it's usually enough for 2 flasks. Well, it's not usually, it's enough for 2 flasks. So I'm going to pack that up, add it in, and then take the next one, do the same thing. And the last one. Cool. So the mitomycin is now in the growth medium, and it's at the correct concentration. So just give it a little mix. And then what I need to do is remove all the growth medium from these flasks. And to do that, we use this. It's like a suction thing, which just has a tube that goes down into a waste bucket down there, basically. So I'm going to take one of these and just aspirate the medium from each flask. So as you can see, the medium's removed. And now I'm going to add 8 mils of the medium containing mitomycin C to each flask. I would do this with one of the bigger chipettes that I used a second ago, but there aren't any more in the drawer just there, and I can't be bothered to get up to go and get some more. So I'm just going to do it 8 times with this small one. So yeah, as I mentioned, once I add the mitomycin C, I put the cells in the incubator for 3 hours. And the mitomycin C will work its magic. I didn't show you at the start, but basically, when you open the hood, well, this is a tissue culture hood, first of all. I didn't explain that either, but this is what you do, this is what you grow cells in, basically. And it's got like a controlled airflow to keep everything sterile. And when you open the hood, you have to like spray everything with Chemgene, and then everything you put into the hood, like what I did with these, you spray with ethanol, 70% ethanol. Okay, so these are going to go into the incubator now, and so I'll catch you in a second. Okay, so the cells are in the incubator. I'll leave them there for a couple hours. But yeah, right now I'm going to close down the hood. I'll clean it, close down the hood, and then I'm going to take you to the other room where they've got like a microscope and a camera, so I can shows you what you're looking down at. How do I say this? You can see what you're looking at down the microscope on a computer screen, so I can show you like what the cells look like and stuff, which might be interesting. And then after that, I think I'm gonna get the Blue Yeti out and do a bit of normal sound assortment stuff. Although I've been thinking, and I think I might film that now, but I might like use it as a separate video, because I figure people who want like a sound assortment video aren't going to want to have sat through like all of this stuff first to get to it, and people who are interested in the like making feeders and stuff might not want to sit through random sounds, so I'm going to split them. But anyway, yeah, so to clean it, you basically just put everything back that you haven't used. So I'll come to use these later. These can be thrown, as can the wrappers. And then that can be thrown, and this can go there. And then you take some ethanol, and you basically need to hold down the suction and spray some, just to kind of clean the tube. And then you take Chemgene, and basically spray the whole thing. And I have some paper towel. Just wipe it. At the moment, our lab leaves all like in other labs I've worked in the past, each person will have their own set of like tips that they put in and out of the hood whenever they're using it. But at the moment, our lab just has one set that we leave in the hood. But to be honest, I'm pretty much the only person doing any tissue culture at the moment. Yeah, if any of you are lab people and wondering why I'm not taking out the tips, that's why. Okay, so I'm going to close the hood. It beeps when I do it, so I'm going to turn the camera off. Yeah, I'll catch you in a sec. Okay, so I'm in the room next door, which technically belongs to like a different lab group, but they have this microscope here, which is pretty cool and much better than the one we have. And that's because, maybe I'll pick you up, they have this camera. So basically, it means you can take photos, you can show other people like what your cells look like and stuff. And it also does fluorescence imaging too, which is pretty cool. So anyway, let me put you back down. So yeah, I'm going to show you the cells right now. So here we have a flask of more J2s. So these are ones that I have growing that I'm not turning into feeders. I'm going to keep these ones growing. And that's because they're at a lower passage. So basically, you grow your cells in these flasks until the flask gets full. And then you basically change them and put them in a new flask. You remove them from you unstick them with an enzyme called trypsin. And then you put them in a new flask at a lower density. And every time you do that, it's called passaging. And basically, so the passage number of cells is how many times they've been changed flasks, and the higher the passage number, the more exhausted the cells become. So with J2s, we only use them up to passage 12. So passage 12 is the last passage that we will use them for feeder cells. Whereas these ones are passage 10, so I'm going to keep them. Whereas the other ones were passage 12, so I just mitomycined all of them. So if I just set them up quickly, there's a bit of condensation at the top, so I will remove that and make sure it's on. There we go. All right, so give me 1 second. I'm going to flip you around. Okay, so this is the screen, and I'm just focusing. And now you can see almost, let me switch to a different filter. A bit dark. There we go. So these cells are the mouse embryonic fibroblast cells. So you can see they're kind of like spindly and very like non-uniformly shaped. So there are some like this one, which is like quite small, and then you have these big ones. You get a mix of like spindly ones and kind of cuboidal cells. So these are quite low confluency cells. So I'd say these are maybe like, I don't know, 40%. And you usually wait for them to get to about 80% confluency before you start, before you want to passage them. So yeah, this is basically what the mouse fibroblast cells look like. And these are the ones that we use to turn into feeder layers. And you can see, like when I move it, there's quite a few things that are out of focus floating around. They're just dead cells. That's normal. So then, let me just take, so these are then human cells. You can see the medium's gone kind of yellow, and that's because when the medium gets used, when the kind of nutrients and stuff in the medium get used up, the pH changes, and that makes the medium turn yellow. So basically I need to change the medium today. And so this is what the human cells look like. So these are human airway epithelial stem cells. I hope that makes sense. But yeah, basically human lung stem cells that I've taken from nasal brushing, so kind of like a COVID test. And then I've grown them, and these are passage 3. And so basically you can see that these are kind of fairly confluent. These are probably 70% confluent. So you can see that these kind of dark, small cells are the human cells, and they form these colonies which basically expand and start to merge. So by tomorrow, all of these colonies, which are currently slightly separated, they will have started to merge. But you can see that in the gaps, like here and up here and here, those are all the mouse embryonic fibroblast feeder cells. So these are cells that I did what I'm doing today. So I inactivated them with mitomycin C, plated them in the flask first, and then added these human cells on top. And the crazy thing is, we don't actually know exactly what the feeder cells do or how they help the human cells, but we haven't been able to like recapitulate the effect of the feeder cells with any other method. So that's why we still use them. But yeah, this is basically what they look like. If I move it around a bit, you can see, yeah, so these ones are quite nicely, oops, accidentally zoomed in. Oh my God, there we go. Yeah, these are very nice, like dense colonies of cells. The cells are really, really small, and then you have the feeders in between. Yeah, so that's basically how I grow these cells. So these cells, I'm making the feeders today, and then tomorrow I will passage these cells and put them on top of the fresh feeders in a new flask. I think that's everything you need to see. Let's move this up a bit. So yeah, I'm going to go back to the other lab now and make some sounds. Cool. Okay, so it's been 3 hours now, and it's time to do the next step. So I've reopened the hood, and I just need to clean it. So it's just the same as when I closed it. Spray it with Kenjin, wipe it down with some paper towel. By the way, I'm sorry if the last section I watched it back. I don't know if I was speaking too like loudly and excitedly, if that's a word. I hope it's still relaxing. This is an ASMR video after all. Alright, so I'm going to get this out quickly from the incubator. So the cells are now inactivated, and so what I need to do now is basically, it's as if I was passaging them. So if I hadn't inactivated these cells and they were growing cells, this is what I would do to change the flask. And that's basically because I want to collect all the feeder cells and then basically put them in the vessels that I want to grow my human cells in. So I'll show you how we split these cells. So this is PBS, or phosphate buffered saline, and that I'm going to use to wash all the cells, because this medium, the growth medium, contains something called serum, and basically that blocks the action of the trypsin, so you want to wash all the serum off. So I'm again going to aspirate all the medium. I don't know if you can see. I hope. It's kind of difficult to get the angles right with the camera, because obviously I can't put the camera inside the head. I forgot to restock the bigger stroppets, so I'm going to be doing everything with a 10mm one again. So you need to wash with roughly the same volume of medium. That's what I do. So I'm putting 10mm. Well, I had 8mm of medium with mitomycin in, but I'm going to put 10mm into each flask. And obviously, the bottle of PBS you want to keep sterile. But as long as when I dispense the PBS, I don't touch the stripette on the flask, I can keep reusing the same one. And I also need to make sure that I dispense it onto the roof of the flask so that I don't wash the cells off. Okay, so the PBS is added. Give it a quick swirl. And then I'm going to remove all the PBS. So the cells are washed. Now I just need to add the trypsin, which I've got warming up in the water bath, so I'm just going to grab it. So this is the trypsin. And normally I would add 2ml to each flask, but I think there might be slightly less than that in the falcon tube, so however much there is, I'll just divide it by 6. As long as there's enough to cover the base, it's fine. So there's 10ml. So I'll just add 1.5 to each. Added a bit more into the last one, and then you just need to shake it to make sure the base is covered. Cool. And then now these go back in the incubator for about 10 minutes. And this will basically, I think it removes like proteins on the surface of the cell membranes that are kind of causing the cells to attach to the bottom of the plate. So yeah, removes these proteins, and so the cells unstick, and you end up having them like floating in suspension. So yeah, but it works faster if the enzyme is at 37 degrees, so it's going to put them in the incubator. And then while the trypsin is working, I'm going to feed these cells that I showed you earlier, because as I mentioned, the medium's gone yellow, so you can tell the cells need feeding. So the medium's also in the water bath, so I'm just going to grab it. You may be wondering why I spray everything other than the like flasks of cells when I put stuff inside the hood, and it's basically just because like these flasks have filters here, so you don't want them to get wet, and these plates aren't really sealed, so it's just risky spraying them with stuff that kills living things. So yeah, you kind of leave it. So this is kind of straightforward. All I'm doing is removing what's in here and replacing it with fresh. The only thing is, from these two flasks, I'm actually going to collect the medium rather than aspirating it, because basically when the cells are in culture, they release a load of factors, growth factors, cytokines, things like that. And these can actually improve like the growth medium. They can help support the growth of cells. And for some procedures that you do with cells, for example, if you want to infect them with a virus, it can be quite stressful. So you can use, we call it conditioned medium. So medium that's already been like, that contains the factors that are released by cells in culture. And it basically helps the cells more, it supports the cells. You have to mix it with fresh medium, because if you just use this is like used. But if you mix it 50-50 with fresh medium, it's kind of a bit better for the cells. But most of the time you just use normal medium. Anyway, so from these two, I'm going to use a 5 ml stripette to collect the medium. And I have this pot here where I'm collecting it all. And then once the pot is full, I filter it with a 0.2 micrometer filter, so tiny, tiny holes. And that will remove any like cell debris or anything and leave just the medium and the growth factors and stuff. I'm going to do the same from this. So that is those done, and then I'm going to add fresh medium. So I'm going to add 4 mils of medium to each flask. Okay, and then in these 2 plates, I also have the same kinds of cells, the same types of cells. It's actually exactly the same patient donors, but with these ones, I've infected them with a virus. So I'm not going to collect the medium from this one, just in case there's any viral particles left in the medium, which I don't want. Theoretically, the filter might remove the virus, but it's not really worth risking it. So these ones I'm just going to aspirate as normal. And each well needs a separate aspiration pipette because you don't want contamination between wells, because each well is like infected with a different virus. Okay, and then to each well I'm going to add 2 mils of medium. And for this it's important that the medium is dispensed onto the walls of the wells so that you don't wash the cells off. There we go. So my cells are now all nicely fed. They're not hungry anymore. And I can put them back in the incubator. And then while I'm here, I'm just going to give the feeder cells a bit of a tap in their flasks because it can help dislodge the cells. I'm going to take one. Just to the microscope over here. I'm sorry that I'm leaving you back at the hood. But I'm just checking in the microscope to see if they are coming off, yeah. Which they kind of are. But they're going to need more time. Sometimes it can take a bit of a while, which can be annoying. Okay. I think that I will cut here and I'll see you when the cells are fully detached and we're ready for the next step. Okay, so the cells are ready. I'm just going to give them a quick tap again. And so basically what I need to do now is neutralize the trypsin with medium. So basically the medium has the serum in it. So the serum will neutralize the trypsin and stop it working, because if you leave it on the cells for too long, then it's bad for the cells. So I'm going to add 3 mils of medium to each one of these, and then I'm going to wash them a bit, wash the flask a bit, and transfer it into a falcon tube. Transfer the cells. And then once I've done that, I can count the cells. So I'm going to take 3 mils and add it here, and then just rinse the bottom of the flask. Collect the cells and transfer it to a falcon. I feel like I can hear someone outside. I'm not sure why they'd be here. It's 6:15 on a Sunday, but so yeah, next flask. There's definitely someone there. I hope they don't come in here. I don't know if any of the people, any of you guys that are watching are football fans, but Man United are currently playing Coventry in the FA Cup semi-final. It's just gone to penalties. I'd really love to be watching it, but my friend's on his way to my house, and I'm still here. Did I say it right? He's on his way to my house, yeah. So I need to finish this quickly. But yeah, it's crazy. Coventry were 3-0 down with 20 minutes to go. Brought it back to 3-3. And they scored a last-minute winner that was like a centimetre offside. It's crazy. Football's a crazy sport. Anyway, almost done. I really have no idea how relaxing this video is going to be. Please do let me know if you have enjoyed it, because yeah, I can always make more, but I thought I'd try it. But it is very different to videos I normally make. But yeah, I guess it's kind of more like background noise rather than, you know, ASMR triggers. Yeah. Let me know what you think. If you hate it, tell me. I won't do it again. If you love it, then I will do it again. I also filmed, it's kind of short, I think it's about half an hour, a half an hour long video during the incubation period. So you can look forward to that. I did like a kind of a sound assortment, but like one of those short attention span videos, so quick changing between the triggers. Okay, so these can now be thrown away. And so all the feeder cells are in here. Give it a mix. And then I'm going to count them using this. I hope you can see that. It's a hemocytometer. So you might be able to see there's like mini grids in each kind of section. And so basically you put some cells in and you count them in the grid, and then from that you can work out how many cells you have. So I'm going to put 7 microliters of cells into the hemocytometer. And then I'm going to take you over to the microscope where we're going to count them. Okay, so the hemocytometer is on the microscope. Just going to focus it, and then I'm going to count the cells. So the grid is like 10 squares made up of 4x4 squares, if that makes sense. So 10 lots of 4x4 grids. And I'm just going to count five of the squares. So 5 of the 4x4 grids. So in the 5 grids, the 5 squares, there's 139 cells. So to calculate the total number, I need to get my calculator and do 139 divided by 5, which is 27.8. And then to work out the number of cells per milliliter, I times it by 10,000, which is 278,000, which means in every milliliter of the medium, I've got 278,000 cells, and I have 30 mils in total. So that means I have 8.34 million cells in total, which is actually not very much at all, considering I had 6 flasks, but they weren't very full. They weren't very confident, so I guess that kind of makes sense. I just need a pen. I'll take you back to the head. Okay, so basically, I'm going to be placing the feeder cells in these 6-well plates because that's what I want to split the human cells into. And the feeder cells have to be at a density of 20,000 cells per centimeter squared. So yeah, I've calculated in my hand how much of this I need to add to each well, and it's 680 microliters per well. So I'm going to do that now. Basically, this one. Just using this pipette. I'm going to pipette 680 into each one. So 2, 3, 4, 5, 6, and then the next plate. 1, 2, 3, 4, 5, and 6. Someone commented on my last video, a funny comment, something like, someone get this man a multi-dispense pipette or a multi-channel, because pipetting all those wells individually was crazy, which I agree. We do actually have a multi-dispense, but the only like attachments we have for it, they're not big enough to do like many dispensers of 200 microliters, which is what I was doing. So I couldn't use it, but maybe I should ask my supervisor if we can get some bigger attachments for it. Getting there. That is 4 plates done. One, 2, 3, 2, 3, 4, 5, and 6. Perfect. And then basically just want to top these up with some extra growth medium. So I'm going to add an extra milliliter to each well. And again, as long as I didn't touch the cells, I can reuse the same stripette. There we go. So that is those ones plated. I need to kind of shake them to distribute the cells, but I'll probably do that after I turn the camera off because it makes a pretty awful sound. But I'll keep these here. And then, so those are plates that you just use to grow the cells. And then these are what are called chamber slides. And so these are small vessels that you grow the cells in, and then you can fix them and stain them. You can use them for immunofluorescence staining. So I'm also going to grow some cells in here to stain them. And that's when I say staining, that means if you want to look for specific proteins to see if they're present, where they're located in the cell, how much there is, you can use immunofluorescence staining, which is where you target the protein or proteins with a specific antibody, and then you can use a secondary antibody that binds to the primary antibodies, and they have like a fluorescent marker attached. I'll probably explain it more in a future video, if that doesn't make any sense, but yeah. So these ones I only need to, so these ones I was adding 190,000 cells per well. These ones I just need to add 14,000 cells, which is 50 microliters. So I'm going to do that now. Hopefully there will be enough of the cells remaining. I used most of them on the 6 well plates. These chambers have, or these slides have 8 chambers per slide. which means you can technically use 8 different staining mixes. So you can stain for 8 different proteins or combinations of proteins. I'm mostly going to be interested in the protein that I mentioned in my last video, which is called P63. And P63 is a transcription factor that is known to be, well, a transcription factor is a protein that controls the expression of other genes, so it controls if other genes are like turned on or turned off in specific cells. So yeah, P63 is known to be really important for the function of lung stem cells, which is what I've been growing. But no one knows what it does yet, so that's kind of the fundamental basis of my PhD project. So I'll be staining these cells for P63. And this is basically part of an experiment where I'm investigating if cell confluency, so how full the flask is, affects the expression of P63. It's not interesting in like a biological sense, but it's important. It's an important consideration for all my experiments because I'm always using cultured cells and I'm investigating P63. So if P63 changes with confluency, I need to make sure I'm then really strictly regulating at what confluency I take cells to use for different experiments, if that makes sense. And then I'm just going to top the medium up again. Almost there. So now I accidentally touched the wall, so it's safer to just get a new pipette tip. Cool. So now I will leave these in the incubator overnight so that the cells can settle, stick down to the bottom again. And then they'll be ready for the human cells to be put on top, plated on top. And yeah, that is how you make the feeder layers. It's very straightforward. It's pretty much the simplest thing that I do in the tissue culture room. But it does take a bit of time, especially because there's the 3-hour incubation. But I hope that you enjoyed the video. I hope that you found it interesting. Yeah, as I said, I kind of have no idea how this video will turn out or how relaxing it will be. I hope it's relaxing. I hope you got some tingles, maybe fell asleep. I think this is the kind of video if someone else made it, I'd put on in the background like while I was working. Just kind of background noise that's less distracting than music. But yeah, let me know what you think. Let me know if you want to see more. Thank you for your support, as always. And yeah, I will catch you. Oh yeah, and soft speaking. Let me know if you want more soft speaking videos. I don't know how soft my speaking was. I think I maybe got a bit overexcited at some points. Even now, I think I'm probably talking a bit too fast. So in the future, I'll try and calm it down a bit. I think whispering kind of always calms me down, whereas parts of this I felt like I was pretty much just talking normally. Anyway, that's enough. That's enough talking. I have no idea how long this video is going to be, but not too long, I don't think. But yeah, thank you for watching. Have a wonderful rest of your day. And I will catch you in the next video.